Hey everyone, welcome back. So for today, I'm going to be talking about our ninth lab for the semester, which is the activity assay. So where we are now, we have purified our beta-gal, as well as checking the concentration of our beta-gal using the BCA assay. So now we really know how much beta-gal we're working with, but we don't really know if our beta-gal is active. For all we know, our protein could have degraded somehow where it got all chopped up. Or more likely, it could have been denatured where the protein starts to unfold and unravel into different configurations. And when this happens, the protein loses its function. So today's lab is basically trying to focus on, is our protein actually active, yes or no? So in order for us to really perform this assay, we need to understand what our protein's exact function is, as well as any natural substrates or byproducts that the protein might utilize. So here I kind of have the example of malate being converted to oxaloacetate using malate dehydrogenase. So measuring the actual conversion of malate to oxaloacetate is pretty difficult. Normally we try to like to use a spectrophotometer, but we can't see this conversion. But we can kind of see the conversion of NADP or NAD plus to NADH. So because we need one NAD every time we convert to malate, we're able to kind of get an idea of how much or how active our actual MDH is. So what does this mean for beta-gal? So beta-gal is a bit of the same. Beta-gal's function is to hydrolyze lactose to galactose and glucose. Problem is, there's no easy way to measure this conversion. If we wanted to use a spectrophotometer, we wouldn't be able to see any of the galactose or glucose. The other problem is beta-gal doesn't need any kind of NAD or ATP or something we can measure indirectly. So if we were to test this reaction and check it on a spectrophotometer, all of our measurements would be clear and colorless, so we wouldn't be able to measure anything. So instead, for the activity assay, we're going to be using a derivative of lactose that looks very similar enough that we can confuse beta-gal into hydrolyzing it. And so that compound is orthonitrophenyl beta-galactoside, or ONPG. The reaction is exactly the same as with lactose, where ONPG will be cleaved and we'll get beta-D-galactose, which will be colorless. But we'll also get another compound called O-nitrophenyl. And when this compound is made, it will actually produce a yellow color that we can measure on a spectrophotometer at 420 nanometers. So for this assay, all we're really testing is if we mix all of these components together, OMPG, beta-gal, and just put them together, will the solution turn yellow? If it does, that's a very good indication that our actual beta-gal is able to cleave and is functioning properly.